Morning.
Yeah, morning, everybody. How's everyone doing? Ah,
Welcome to Microbiology Twitter Journal Club.
yep. Welcome. Hello. My name is Fazal Lam. I'm a former microbiologist, research integrity specialist, and publisher, and I'm joined by my colleague Danny Chan, who,
Yeah, my name's Danny. I'm also a former microbiologist, now fact checker in the pharmaceutical advertising industry, and a community member in DIY biology.
yeah, and
And running this series of journal clubs over the past few weeks, I guess, because of course, the event that revive interest and talk about papers critically in the public eye. And so we've been choosing papers that are related to the SARS-CoV-2 virus.
yeah, mm,
And we're speaking about um, digging into the figures. I think that's our focus. Our basic is on the off weeks we choose papers from, and then uh, on the on weeks we dig into or what we'd like to see or what we're learning and what we're learning ultimately. Yeah, because papers are an interesting window into the mind of a scientist, and not a lot of people get to engage in the primary literature. So we're hoping we can help people through that. We have a discussion. We have a hashtag on Twitter, hashtag microtwjc. So if you have any questions in the course of listening to us speak, please tweet them out, and we'll respond to them as we can.
yeah.
Does that sound good for the front matter?
That's all good. And yeah, so shall we talk about the paper we chose? So, Inovio's DNA vaccine.
Yeah, and
Yeah.
this was a preprint when we chose it. It still is a preprint, right? I didn't see it get published at all.
Yeah, no, it seemed to have moved away from just being a. It wasn't just. At the moment when I
Oh,
started, it was said it was accepted,
right,
and
not
then
a strict print. Under review, it says.
yeah, now it's under review, and which which inspires confidence. I feel,
Yeah.
but I mean, the interesting thing about this paper is because I remember last week I told you about there's a strange paper where they're electrocuting people and giving DNA, and it was this paper.
That's right. Yeah, we were talking about electroporation. I was like, in people, that's so weird. And then I read this paper during the week. I was like, oh, it's it's a real thing. It's interesting. I was reading the abstract of uh, it's a paper where they work on like the adjuvant strategy, I guess, in this intradermal electroporation. And they actually say that it would be really great. I don't know why they say this, but intradermal vaccine style of vaccine, they imagine it being easier to distribute, even though you would think that you also have to shock people in conjunction with it, which is kind of bizarre.
Yeah, that I mean, I really got shades of starts. Is this how we end up in a dystopian like landscape?
Yeah.
Uh,
And that is how it's done. That's how this vaccine is administered. Um, the idea is that they don't have to put it so deep into your muscle or anything like that. But then also the fact the electricity allows it to get in there a bit better. This is like a really
yeah.
common research tool, right? Where
Oh yeah.
yeah, this is like the primary way of getting DNA in the research setting. And here they have like it's a weird like pen-like device and like an array of needles as well. So like, I guess the idea is that it'd be like a patch. I think that that's what they're probably hoping to. I don't think it's there yet, but the idea of
Yeah.
like a patch and then some electrodes to shock it into the skin.
Yeah, so like that's the kind of thing that scares me a little bit. You've got these electrified needles that you're sticking into people, and the way they do it is they do like a kind of initial shock, they inject things and have an initial shock, and then they wait 30 seconds for the second shock. Now,
Hmm.
when you've got a needle in you, that feels like a very long time. That feels like
No, but the needles are not very deep. like
Mm.
only into the like I don't
Yeah.
think they're that deep. Like in this, I have like a in Sardasa 2011. Anyways, there it's the one where they talk about the technology. It's called they TM'd it. This is the company, right?
Yeah.
Selectra. um, yeah, I think they only go in like up 20 microns maximum.
Yeah. I mean, they
It's
did do
it's
a.
not very, it's not very deep.
I did come across a study where they looked into it and there was and they asked and and they uh and they basically asked people, oh, how much does this hurt? On a scale of 1 to 10, being 10 being the worst thing you've ever
Oh,
felt,
and
and
what
they
did they
was
say?
about a 7. They gave it.
Oh, oh, so it is kind of
It is
painful.
kind of painful, so it's not ideal by any stretch. Uh,
Sure. But yeah, so this the reason why they have to do this is it's a DNA vaccine that they're talking about. Um, so DNA vaccines, obviously, like this is a RNA virus. There's actually no DNA. I don't think there's any DNA produced
hmm,
in the course of the infection
no.
with the novel coronavirus. It's just RNA. It goes into the other strand of RNA, and then it goes back to the RNA of its genome, positive or positive to negative to positive.
Yeah.
Um, so a DNA vaccine is going to it's designed to make a protein,
Yeah.
and it's a protein that the body will hopefully do.
Yeah. And
And again, so I was gonna say, and it's not new. It's not like brand spanking new technology. It's something around for a while. In fact, the previous literature on this topic is talking about them using it for a MERS vaccine.
yeah.
In 2004, they put it in mice, and then as in 2019, it's in clinical trials, or they're reporting the clinical trial,
Yeah.
the phase one clinical trials for that MERS vaccine. Um,
Yeah. And like electroporation itself has been around. So I know I used to do it in the lab on bacteria, but I
yeah,
mean it's kind of interesting part because
yeah.
yeah,
I mean,
when
it's done in more than bacteria. I've also done it in cell culture. I've seen it done in cell culture, and I've seen it done when I was working in the mouse lab, like a brain research lab. They used to in plasmids into the liquid cavity of the brain,
hmm,
right? There's like a space where liquid is in the brain, and they would inject the plasmids into there, and then put tiny little paddles across the cortex, and then shock it into the base layer of cells there. Because like in the brain, I guess it's kind of like or a lot of development. It comes from a basal layer of stem cells, so it's a
oh
way to get DNA into those stem cells. So it's not so strange in that sense, right? Like in the research setting, it's there, and this company obviously has put it in putting it to humans.
yeah.
Yeah.
So yeah, I mean,
There are other DNA in though to a cell. I
oh yeah,
Yeah, this is just one of
yeah,
them. And, it just happens to be the company who's done this research chosen to pilot out the way of doing it.
yeah. I mean, it's because a lot of the other like require other viruses or like maybe some certain chemicals. So theoretically, this isn't as bad. It doesn't input new chemicals in your body. It just gives you a nasty shock.
Yeah, yeah. When
So
you're thinking about like the safety objects, in some ways, it's only the DNA that you're sort of evaluating in the scenario, and then the medical
yeah,
device. You don't have to worry about a biological agent. If in the case of like vaccinia virus is how they would deliver that,
I mean,
or
there's
one
like
of those DNA-based viruses.
Yeah.
Um,
I mean, chimpanzee adenovirus is one thing they're using for another vaccine, or like
is
various other like.
is chimpanzee adenovirus a DNA virus?
I don't know. I don't know. Maybe I don't think so. Maybe I.
Okay, I don't know. We'll have to
That
look that.
Yeah.
Something to look up.
I'm gonna say no because yeah. I mean like yeah. The vaccine virus definitely is a DNA virus. I remember learning that
Yeah,
in undergrad. But yeah.
yeah. And then I guess you can do liposomes, but it's more common, at least from what I know of the RNA vaccines out there. Actually, I was looking at I think I even saved it down to our Zotero group. We should mention this actually at the beginning of our things. We're trying
Yes.
to keep track of some of the papers that we read journal in a citation manager called Zotero, and then we have a public library that you can check out there. So if you go to our website, I think you should be able to find that. But I
Yes.
just want to say that I found a doc from the Y-Star Institute. Have you heard of that
Yeah.
organization
Yeah. I
before?
heard about them. Yeah.
And,
They.
and they have a listing of all the vaccine. I think they're trying to keep a up-to-date list of these vaccine candidates. Um, and so the DNA vaccine, there's like a very long list of DNA vaccines. Oh, they're also doing treatments, both treatments and vaccines. Um,
Yeah.
and it's a great list, and you can see. And then they also say like all these different ways. Like there's DNA plasmid needle-free delivery. There's some
Yeah.
different ways that they're putting the DNA in. Yeah, lots of different ways. I don't see any ones that say if they're delivered by bus.
I mean, the big Oxford studies were chimpanzee adenovirus.
Okay.
Um, so, uh, yeah, shall we get onto the paper?
Yeah, yeah, yeah. I guess we were talking about some background. I mean, that's the background of that. The other thing is talking about the animal models they use. I guess we'll get to it as we go through the figures.
But I mean,
So yeah,
we can talk a bit about that now. I'm just preparing the next like kind of overlay image and trying to get everything in order. So, uh,
okay. So they look at two. They're actually looking at two animal models in this paper, uh, mice, and then they look at guinea pigs. Um, and mice, just because they're easy, we talked about this last time. We'll
yeah,
probably say it over and over again. Mice are dirt cheap in compared to other animals. The techniques that are established for them are very well established.
yeah,
Schools or research institutes have colonies of mice
yeah,
in their basements usually,
yeah. I
and
mean,
yeah, you just order them from one of these two labs, Jackson or Taconic. I think they use them from both those
yeah,
major breeders in this one. So those are two American-based um,
yeah.
breeders. I don't. What do they use in the UK? What?
I mean, we also use Jackson Taconic. They've got European kind of
Ah,
places there.
okay, yeah.
The, I mean,
Multinational
it's
corporation.
yeah. I mean, they have to. I mean, otherwise you have to fly a mouse halfway across the world to get to some places. I mean, what usually happens is like you go to an institution and then they establish their own breeding colonies for these different mice,
Yeah, yes.
which can be an issue because for certain studies, you get different results simply because you got different mice who've like had genetic drift and had different mutations. But that's me moaning about something for another day.
Yeah, so they used the Charles River and Jackson Lab
Yeah,
mice that they used. And then they also had guinea pigs, which I guess they're better than we talked about this briefly last time too, right? That
yeah,
mice they get like a really high fever or a high fever, or like their infections kind of different than humans.
yeah,
And guinea pigs, I think, at least the symptoms that they get, especially when you're talking about fever, it more closely matches human beings.
yeah. Um, I mean,
uh,
as animals go, yeah.
yeah,
Yeah,
as non-human primate, as non-primates go, I guess. Um,
yeah,
and it's also about their respiratory system. I'm pretty sure that the guinea pig respiratory system is a little bit different. The types of immune cells that line their airways.
yeah. Uh,
So yeah, so then they inject these. So the vaccine will be delivered by this electroporation system into
yeah,
these two animals, intramuscularly, actually.
yeah. Well, the with the mice it's intramuscularly into one of their leg muscles, which is actually quite a deep and quite a painful thing to do,
Right.
because and with the guinea pigs they did it through a man-tooth injection, so into the skin. So I don't know if you ever had tuberculosis like skin test, where you had these
Oh,
like just
yes. Yeah.
multiple needles going into your skin.
Yeah. I remember.
Yeah,
Oh,
imagine
I
if it
think
was.
I think they're done a bit differently. At least the one I had, it was just like they kind of lift it up and they put like a bubble of liquid underneath my skin, and
Oh,
then that just kind of absorbed into me.
with
Yeah.
with my one, it was like they had this like really scary aperture rates with like all these like needles, and they went poof, and it's like what? Oh, it
Right.
wasn't as bad as I thought it was.
But that's
And like
similar
so,
to what they would have done with the guinea pigs, right? Like needle array and then followed up by the electroporation device.
well, I mean that's what I presume, but they say in this paper that they did the Mantoux test, but that's like a very specific test that relates to tuberculosis antigens, which would
Hmm.
be very weird if they did that in this study. So what I assume is that they mean they used the same kind of needles for
Yep.
that as guinea pigs.
Yep. Yep. Right, but in the first figure, so that's and they end up going through and they study the immune response in both of these animals. And I guess we'll get to it later, but they don't really ever show any efficacy, right? As
Yeah,
it relates to as it relates to an infection. They're just showing us like what the immune response looks like, but
yeah,
not the functional outcome of whether or not it repels infection in these animals.
yeah,
But before they do all of that, they have to make the or they describe to us how they've made their
yeah.
construct.
How are they doing?
So that is yeah. So that
So
is figure one.
yeah, so I've put up figure 1A at the moment. So figure one basically like kind of shows that they looked at a bunch of different coronavirus antigens and tried to find the one that represented the broadest amount, I guess.
Yeah.
Um,
And they had done this from what I read in the paper. They had done this work before the 2020 or like 2020. It is 2020 before the
yeah,
most recent cryo-EM structure had come out of the COVID-2 protein. So they were
yeah,
doing this based on early sequences. They aligned a bunch of sequences and they're like, what's the most similar sequence here? Um,
yeah, and
common sequence. And so in A, that's the alignment. In 1A, that is the alignment that they're doing.
yeah,
Um, so amino acids, and they show the I guess the domains of interest in red.
yeah,
Receptor binding domain, so that's going to be the thing that grabs onto the
yep,
ACE2 cleavage site. I think people
yep,
were saying it's cleaved by furin, but perhaps other proteases will cleave that. That is to activate the whole protein. Fusion
yep,
peptide is like something that gets
yep,
into the cell membrane and fuses the membranes together. And transmembrane domain, that's what's going to anchor it onto the surface of the viral envelope.
yeah. So these are all like really important like functions. I mean, these basically are what help the virus work. Um,
Mm-hmm. And I
so I
love.
mean, I've pulled up. Sorry, you go first.
Oh no, I was just gonna say like when we were choosing this paper, I mentioned that like this is a really nice figure. We haven't seen this yet, at least on Micro Twitter Journal Club. Just to see those three spike proteins in the membrane, you can see that membrane is made of so many phospholipid molecules. Where you're like,
Yes.
it's just like a cloud of dot, really. But the green is there. That's the hydrophobic section of the lipid bilayer, right? Those reds are
Yeah.
presumably the uh, positively charged phospholipid heads. Um,
And
yeah,
and
go.
and you got like the proteins. So I've pulled up the proteins themselves, so you can see like actually the structures like seem quite similar, but then they differ kind of in the receptor binding domain, and it's kind of interesting that because again they did all this work, even though they didn't. I mean, they did all this work from scratch because everything was done simultaneously.
Yeah.
So yeah, it's kind of impressive that they went through all this. So I mean, I think I mean in the SARS-CoV-2 they do highlight the receptor binding domain up in yellow.
Yeah, and you can roughly see too from just like if you zoom in a bit to the graphic. I'm zooming in right now on my PDF, and you can tell that yellow part looks different than the corresponding lump in SARS and MERS.
Yeah.
Right? There's like
So
all these in MERS. There's tons of these beta sheets that are
yeah,
really easy to see. And like that's not the case here, and we know right MERS has a different receptor entry point, so it
yeah.
makes sense. They use a different so the receptor binding domain.
So
Uh,
yeah, it's really nice to see that. You can see
yeah.
like so viewers will probably be seeing like 3 by 3 kind of like straight lines in the MERS, but in
Hmm.
the area that would be yellow. But So I
Exactly.
mean,
Exactly.
yeah, and it's
And then there's
kind
even
of
some differences when you look at between SARS and SARS-CoV-2. Like I can see there's like a funny red arrow pointing down in SARS. That's the arrows. The flat arrows are the beta sheets, and
yes,
then the helices. Well, they look like helices, but that structured section doesn't exist in the CoV-2, at least in the model they have here. And again, like I think important to remember too that these are like these are just the whatever information they had at the time to generate these. This paper didn't do it. They pulled it from a database, probably the you know RSMBD, the protein data PDB.
yeah,
So this is what they had at the time, and this is sort of what structural biologists create. This is their domain. Uh,
yeah,
Seeing the
um,
differences in the shapes helps us know like what's important in the amino acid structure to make. And so like because they have to create a synthetic piece of DNA that is create these proteins, they have to make sure that they're choosing the sequence that makes the protein that looks most similar to SARS-CoV-2. Um,
yeah,
that would be in circulation, right? I think that that's the other part. It's that like they're making it now with some, what they saw at that time, but then they want to make sure that it's relevant. Okay, I noticed this was a weird thing that I noticed in the text here. They said
yeah.
that they optimize things for immunogenicity.
Oh yeah.
I don't
So
understand that. That seems weird to me. How do you optimize a DNA sequence for immunogenicity?
this goes into a bioengineering thing where so I don't know if you've ever heard of gene optimization where essentially you've got tRNAs so the genetic code is kind of redundant. There's lots of tRNAs that do the same function,
Right.
but different cells have different amounts of it.
Yeah, so
So
you have to make sure the codons are optimized. That's I get that from the enhance expression, but then they say enhance expression and immunogenicity. And so like
yeah,
that's I understand like high expression potentially leads to high immunogenicity, right? But in my mind, I think that that's a bit because they are talking about a proprietary in silico gene optimization
yes,
algorithm. So I do think that the tacking on of immunogenicity there is part of the stretch. So this is very much like
yeah,
my job function when I'm checking things for like you look to papers to say like are you allowed to say something like that and be written here because they stretched it a little. Like really what they're saying is they want lots of protein and lots of protein will make an immune response, right? There's nothing unique to that. Like enhance expression enough statements in this sense. Um,
yeah. So yeah, there is no justification for really saying that because well, everything's proprietary. So all you have to do is assume. They don't really give you the idea of why that is.
why that would be, yeah. And enhance expression, from what you said, co-optimization, that's perfectly by the book. Like that makes a
Yeah.
lot of sense. That's how you get lots of protein. But immunogenicity, I mean, I wrote a note for myself. It's possible that somehow they tailor the sequence so it's expressed in a certain like time or in certain cells. That could
Yeah.
be like what they're talking about. But again, like not that is we can't comment on it any further, except to say like it doesn't make that much sense. They do say that they put it under E secretion. Leader sequence of IgE is
Yes.
used.
That is
So
an interesting
that's
one.
yeah. I
'Cause-
was reading through that. It's common. That's one of the peptides that is often used for mammalian proteins. Um,
okay. I mean, it's interesting because like that's more associated with the allergic, and so of the immune responses, there's some that aim towards viruses, some that aim towards like external like invaders, like
right.
alkaline or like, and IgE tends to go towards external invaders rather than viruses, which is kind of interesting to me.
Yeah.
But I mean,
Yeah. I mean, if there's any immune response directed to that leader, that seems bizarre. But, it's only I think they're only putting it so they can get the out of whatever cell ends up getting into.
right, okay,
But from the mechanism side of like thinking about these things, like we're not going to end up talking about any of this during the rest of the paper, but like I'm always wondering like what cell type does it get in when they shock your skin right, with plasmid, right? What's the important cell type that this plasmid has to get into to generate the
I
immune
mean.
response?
That's $1 million question
Yeah,
because they
yeah,
don't really because they something that isn't really touched upon in this paper, and I feel it should be like We do want to
Right.
see like how well these things are expressed in vivo.
Yeah.
Um,
I mean, I think it should be, but I also think that this is also very typical of vaccine, right? Like when you know that a vaccine methodology leads you to antibodies, you don't need to open that up anymore, right? You don't need to
yeah,
question that necessarily, right? Like that becomes your experimental like lens in which you do everything, right?
yeah.
Is that like this produces antibodies? Let's find out. Do those antibodies do something?
Yeah, I mean, because that's your job. That is like beyond your pay grade, basically.
Right, right, right. And I think like it's easy for us as like sort of armchair scientists, right? Like, oh, I want to know more. But like obviously, there's always constraints on this sort of stuff. They're driving
Yeah,
towards a specific point.
I mean, yeah, I feel like it's a job of every scientist to say, I want to know more. It's literally our
Yeah.
job title.
Totally. Yeah, so they end up so that's the DNA construct they do in 1A and 1B. They're showing us like what they hope to be making, what they
Yep.
put the plans in for. And then in the next figure, um, they're showing us the actual the construct maps. Yeah. And then they
So,
put it in cells and they do RNA RT transcription.
yeah, reverse
Uh,
transcription kind of a race to see how something can make lots of RNA faster has
yeah.
more, basically. So that's why
Making
those.
They're making RNA.
So,
They're
yeah,
both making RNA.
yeah, they're both making RNA, except the blue one seems to be losing, unfortunately. It doesn't seem to be producing as much.
Yeah.
Um, and yeah, they just that and the gel is again that shows you the RNA glowing directly. So
I think the
that's
C is
is it.
RNA. No, I think it's a Western blot because they have
Oh, it's a Western
actin.
blot.
Yeah,
Oh right, yeah. In that case, that shows you that the
actin. The
protein's being
protein. So they're
yeah
they're covering all their bases here.
yeah
Once they are able to put these, and again, this is not in the animal yet. They're still using hex cells. I think they're using
yeah
I think they're
yeah
using hex. Yeah, 293 cells. That's a
yes
transformed human cell, like a very
yes
old thing that's existed in the lab a long time.
yeah
um,
it's
and at least in that model, they're able to get um, RNA and protein, so that's good. And they the protein in D with with fluorescent, right?
yeah fluorescent looking at the cells and trying to, and showing that the protein is expressed. I
Oh
mean
yeah, they in the cells. They're using an antibody against the spike protein.
yeah.
Yeah.
Again, that seems. I mean, these kinds of images seem kind of messy to me because the color is quite difficult to tell whether that what's negative, what isn't, because the all I can see is that there are less cells. So maybe I'm interpreting that wrong, but
No, it's the green, right? Because they use some sort
the
of
green.
antibody serum against the spike. So if you see the development of the green color, it means that spike protein is present there.
Right. Okay.
And then the blue is DAPI, so the blue is nuclei. So these are like really dense, confluent, sets of cells, and then you see like each of the cells have these. I mean, I guess it's kind of, this is not that important from the vaccine standpoint, maybe from a clue about expression. Why would the expression be punctate?
Hmm.
Right? Why is it all the? Oh, that's maybe like because they don't get in a cell. Like
Yeah.
each punctate thing is a cell, so like they get it into a bunch of the cells, but not all. Yeah, I don't know. This is very like this is just lab bench stuff to make sure that all their stuff is working, right? And it's
Yeah.
not. The
Yeah.
point
This
is,
is stuff. Yeah. I mean, this is the stuff where it has to go right or wrong, basically. And like you have to make sure everything goes right because if there's a fuck up, you have to find out as early as possible. I know that
yeah,
from my
it's
experience in the lab.
it's a waste
Yeah.
of resources to go
Yeah.
further, right? If you go in and then if you say the first time I'm going to test something is when it's in an animal, well then you have your cohort of however many animals, 50 animals or something. You've injected it all and it doesn't work,
Yeah.
and
I
you
mean,
don't know
like,
why.
yeah. I mean, if this was in my lab, these would be the two most best of a whole raft of plasmids that didn't work, and
Oh
we
yeah,
were, yeah,
absolutely. That's another good point to point out. Papers, right? Is that like behind the scenes,
yeah,
scientists are trying many different things. There could have been more of these two plasmids. Certainly, to make
yeah,
plasmids and transfect the cells, that's not a lot of. I mean, sorry, I don't want to say that's low effort. It's effort. It takes a lot of work, days.
hmm.
months, right, of working, perhaps, to get the results for a whole panel of them. But I mean, a lot less work than animals. So it's definitely not inconceivable to think that they did 10 plasmids and they're showing 2 of them.
Yeah, it's kind of standard. My entire first year of my PhD was electroporating plasmids into cells. Cells like later. So, I mean, it's a lot to get those results. Then you can move on to the next thing. So they, let's see what was on. So figure 3, where it was IgG binding string for basically. Oh yeah, so these were after they injected, after they gave the mice the vaccine, they took serum samples and checked to see what kind of immunity there was for that.
Mm-hmm. And by immunity they mean an ELISA.
Yes.
So ELISA, what? Oh my goodness, I haven't had to discuss this in a while.
Yeah, for immune. So yeah, ELISAs are based
Immunosorbent.
Enzyme linked in the immunosorbent assay. So
I was like, what is that E? Okay.
Yeah. So I mean, to explain what. So an ELISA plate is a plate that's embedded with like an antibody specific or something. In this case, it was for mouse immunoglobulin G.
Mm-hmm.
And I think there's like ways to, and using the very specific protein. So they use a number of different versions of the spike protein from SARS-CoV and SARS-CoV-2.
That's what they have in the legend, right? They have all these different versions of what they're using. Some of it is just the spike, but some of it is just the receptor domain, I think, right?
Yeah.
RBD. And then they have different versions of it too. Just the C terminus, just the
that's so yeah, they basically had, and different parts of, so I mean they had C terminus for SARS-CoV but not for SARS-CoV-2. There was like theory in vaccinology that I think the C terminus always pokes out all proteins, so if you tend to put towards them, that'll always find something. At least I worked on a protein that was on a similar idea, but I'm not sure exactly how well that turned out in the end, because it's been. But I didn't really focus too much on that during my PhD. What is interesting is the controls for, so I mean for like proteins that like S1. So the original version of SARS seems to have some background immunity in some of the mice. I don't know if it, because like some of the bars seem
immunity. So like I guess like the way I like to say it was cross reactivity, right? The serum has cross reactivity. We immunized with one protein, but upon taking the antibodies from that immunization, we're finding that it also cross reacts with another protein.
So
a little bit. It's not a lot. It's a little bit in this one. Yeah.
I mean, that's interesting. So, like, it basically seems to show that SARS-CoV-2 there is a protein reaction to these different parts of the protein, and with SARS-CoV-1, if you just looked at the first one of those graphs, you'd think that, oh, there's a small reaction, but once you take it in context with the control, you kind of think maybe that's just background.
Yeah, it might be even less. I mean, the fact that the SD is like much wider on the serum, right? Like, because I mean, to me that says that there is something going on, right? Like it's within. Yeah, I guess I don't know. It's also within noise in some ways. I mean, but they're so tight on the control, and the control is still serum.
Yeah. I mean, it's I mean, again, I don't like graphs that don't show both like error bars, but that's just my own little thing. But I mean, it does look like there is a definite difference, especially when they do different. So when they dilute the serum. So actually, I don't know exactly why they showed that, but it seems like a good thing to show.
That's an ELISA thing, right? Like ELISAs, they're only sensitive to a certain amount. Just sort of, it's to help visualize better, right? Like what the strength of the binding side of each of them.
Oh right, yeah, of course. There's like that, just getting the right threshold to see everything. So
exactly, yeah. I don't.
So yeah.
Again, it's kind of it's a bit arbitrary here, and you'll see like in the later ELISA, do like a full span of dilutions, right? Like that's more typical when you're doing a full ELISA. They're just screening here, just to see what's going on. And even say IG binding screen of a panel. It's not like they're not meant much in-depth information at this point.
Yeah, basically, from what we can see here, is we can see the amount of well, we can get a guess of the amount of immunoglobulin G in that's specific for these specific proteins
Yeah.
in like kind of the mouse bloodstream.
Yeah, in comparison to each other, I guess, right, is the most helpful thing. So no,
Yeah.
there's relatively more like these antibodies that are produced in the serum. They're more of bind specifically to SARS-CoV-2 spike protein than SARS-CoV. Although there is seemingly some binding, some cross-reactivity.
Yes. I mean, yeah, that's the interesting thing.
Yeah.
And oh yeah, so the next one, so I'm looking at 1A, which is, I mean,
4A. Yeah.
Oh, sorry, 4A. Yeah. Uh, yeah. So that's looking directly at the receptor binding domain antigen in, yeah, in biopsy mice that were injected intramuscularly.
Yeah.
So this is actually hold on. Oh, right. I remember the figure legend annoyed me a lot because some of the figure legends are labeled a bit interestingly.
Yeah, it's kind of confusing.
I mean, like the actual like graph values, like they are listed somewhat differently, and it's kind of frustrating. So the axes change. I mean, this is all minor nitpicky stuff, and this is not very relevant to how good the. It's just like kind of the irritation that I get when I'm reading a paper.
Yeah. I mean,
yeah.
you want things to be an easy reading experience, and I guess
Yeah.
your editor eye is coming out here, right? And being
Yeah.
like,
It's
is
like
this the easiest to understand?
Yeah, this is kind of probably you want from a preprint. I mean, you want to see these sorts of things in a preprint because then you don't want to see like people faking data because you want to see like kind of it's not fully done because they're in a hurry, so they
Yeah.
did mess up with some of the formatting. That's fine.
Yeah, so like it's weird that they do A and what's the difference between A and C and B and D? It's it, the difference is that A and C or A and C are
different things in different parts of the paper.
Yeah.
So like in the figure legend, it's not especially clear, and in the results section, I think so they both say different things. So I'm trying to figure out which one is which.
So A and B is one, and C and D are another group of mice. That's so when you read this horizontally, those are two. We're looking at two experiments, two groups of mice. So that's the horizontal read, and then on the vertical read, we're looking at
the endpoint titer. So the kind of the amount of antibody you have at the end of the experiment, I guess.
Yeah, but whether or not we're looking at, it's like what they're using. Are they using the S2, like the spike protein, or are they using the receptor binding domain protein?
Yeah. Again, that's
B and D. Yeah. So B and D, they're looking at the receptor binding domain, and then A and C, they're presumably looking at the.
Right. Yeah. It gets the point across. It just makes things a bit confusing to read, and can sort of. So I can.
To me, I don't see why they couldn't. Maybe because they want to show the p-values between the endpoint titers, but technically it's the same. Like it's the same data, right? Because it's because endpoint titer is it correlates to that log serum dilution factor. It's when the dilution factor reaches so low that it's a detectable limit of the ELISA that the color disappears, right? And OD 450 is that color from the ELISA. So you see, like as you dilute the serum, the color of the ELISA goes down, right? By the time it reaches undetectable, that's your endpoint titer. So they just rearranged then the axes. And so they could show us just the one data point and the p-value that comes out from it. There are better ways to show ELISAs, I guess, or less confusing.
So we got like it's but and I guess what they're trying to show is that
what we see is the two levels of vaccine, you still get robust humoral immunity to both these. Both the entire spike and the receptor binding domain.
Yeah, and I think they want to show like a dose response as well. So mice reacted with 2.5 micrograms of the DNA versus 25. I feel that they're trying to show that okay, adding more is better. Although it doesn't look
Yeah.
like it's by that much, but that could just be from the because of the log values. But
Hmm. Yeah, I mean, I think those curves would be considered different. I mean, there's overlap in the points. It's clear that through the dilution, like this is what you ELISA curves, and people say this about CT curves as well.
yeah,
Sorry, just like jumping back up
oh yeah,
to 1B. Like
yeah,
1B is how the diagnostic test, right, for novel coronavirus works in some ways, right? They're looking at transcripts, right? Oh,
yeah.
that's dosing though. Never mind, I take it back.
Okay.
I thought that was like a cycle number down there.
I mean, I think that is a cycle number though, isn't it? Let me. No, no, yeah,
Mm,
that was dosing. Sorry, yeah, I
it's
remember.
cycle number against dose. Like
Yeah, that's right.
technically, it's like when you actually do one of those tests, right? Like you're seeing a log graph appear, time, right, and the amount of copy.
Yeah,
So this is sort of a similar thing, like time in that reaction. It's just like developing, but here you have like a sample, you dilute it down to create that resolution, so that you can see the differences between these curves a bit better. Yeah. Anyways,
yeah, because yeah,
let's move. This
yeah. Let's.
this is more just like this is all about that ELISA test, right? That
Yeah,
antibodies are being made and that they're being made specifically to a thing. Could be that giving more of the vaccine gives more antibody. We'd probably
yeah,
see another variant of that. Because it's a tenfold difference what they give, but oh yeah, the log does make it confusing because is that a
yeah,
tenfold difference between there? Yeah.
yeah. So we move on to the next one, which seems to
Figure six.
fig, figure five, I believe.
Oh, figure five. Yeah, figure
Uh,
five.
yeah. So where they're
So now
looking,
they go into guinea pigs,
yeah. So looking at the tests. So the
and
terrifying
they do
multi
this,
needles.
and they do the same thing. So like again, just like we're this whole paper is set up like the progression you'd expect to see, right, in preclinical animal models, right? Is kind of like increasing complexity of the model, getting closer and closer eventually, right, to the phase
Yeah,
one clinical trial. Oh, which by the way, the phase one clinical trial of this particular compound is going on right now. It's
yeah,
recruiting. Yeah,
yeah.
it's recruiting.
And that's
Um,
why I selected the paper. I was like, they're electrocuting people in the face. What are they doing? What are they doing? They're giving people plasmas. People having superpowers. People like Bioshock.
Yeah, I mean,
But
we could
yeah,
pull those. You could pull those study designs and see exactly, right? Are they using this matrix to deliver it intradermally? I would guess yes, because I think that that's the whole point. Like, want to give they want to give a shallower action.
yeah.
Yeah.
And I get the impression that they want to like get the anti-vaxxers and the 5Gers to come together in one mass conspiracy. Oh god, like a. Oh.
uh, so in five, they're basically eating the information that we've seen in figure four
Yeah.
and three in guinea pigs,
Yeah.
and showing that they too. This time they're showing us curves from day 0 and day 14. Oh, I see. Because
Yeah.
they have a
So.
smaller. It's before vaccination and after
Um.
vaccination. Or like,
Yeah. I
yeah,
kind of feel that this is a much better tool. I mean, also using less animals generally is better, and so I do like the day zero and versus that makes sense. And they compare that with the empty plasma, which is just DNA that doesn't have any antigen on it, which is another good thing to compare. And
yep,
then they show like, I mean, it's one of those graphs that would you say is significant even if you do any statistics because the SDs don't overlap and it's
absolutely.
yeah, it looks so that
Within
looks
within 14 days, there is a response.
yeah. So yeah, they definitely respond to an antigen, which is good. That's a good start. I mean,
And then B is again, they just reformatted the data from to
yeah,
make something that they can show you a p-value for.
Yep, because you need that p-value, otherwise people won't pay attention to you. It's called the pay attention value. Should I pay attention to this or should I not?
Right.
Like, it needs to be really small. You need a really small
But
p.
yeah, also like I think this is like I felt a lot of this pressure when I was in grad. Like, yes, that's the pay attention value, but it's also not the value in which you base your understanding of things on, right? Like,
Yeah,
that's pay attention, but that's like one evidence to say that this is different. But like, what else
yeah,
is different, right? Like, you're not just studying the difference between two values, right?
yeah.
You're studying a phenomena, a complicated
Those values need to
phenomena.
be,
And so
yeah,
again, yeah,
yeah.
it needs
Those
to mean
values need
something.
to be meaningful. Sorry.
Yeah. Okay, so moving on. I think this is I had an exciting or an
Yeah,
interesting graph to me. I guess it's actually the next two. This is like because I'm obsessed, or like right now everyone's talking about antibody tests, or everyone
yeah,
imagines antibody tests. And I was saying to someone on the phone recently that I think that people are hoping that positive antibody test means they're safe, right? I have
yeah,
antibodies, so I'm safe. But I think people also forget that like that just like how one of the criticisms of this paper is they never an animal and show that like the infection can be repelled by these antibodies. This is another thing, like just having an answer for yourself and showing that you have antibodies. What does that mean in terms of your ability to repel that disease? But more importantly, I think your ability to transmit that disease, because
yeah,
you have antibodies and you're not going to have a bad outcome when you get infected, but that doesn't not going to have replication and shed.
yeah. So,
Right? So like that's there's a big thing, and like it's going to take a lot of data to get to the point where we can start saying that this positive antibody test means that you're okay to go and do the same things you used to do. Maybe we'll never get to that point, right? Like we may get vaccines before we get to that point. And so, anyways, I just want to say that here, and
yeah,
that's why I'm fascinated about learning more about what do these antibodies actually target, and sort of distantly to me, I wish that we could have paired data with also what's neutralized, right? Like now that
yeah,
we've characterized this pool and we know what epitopes exist, now I want to know what and does that neutralize the virus?
yeah.
even
Well,
even a simple test, and again, this is going to be difficult because I think it's difficult to work with live SARS-CoV. Like in this whole paper, they never work on the live virus, right?
yeah,
Like it's a limitation of this lab, right? That they can't do this work. I would have loved to see these pools that they've characterized so nicely just in a simple like in an in vitro infection test. Just like mix it with some virus and infect some cell lines, right? And
yeah,
that would just be that little extra bit
yeah,
linking the epitope stuff to a functional.
yeah. So,
But anyway,
like, quickly explaining what they're doing here, they took out some splenocytes. So this is what essentially looking at the T cell response, like correct. So they took out some splenocytes from the mice that were infected, and
yes.
then they took like the proteins from various and they chopped them up into small pieces and like about like 15 amino acids, basically the same length of amino acids that. So for interior cell responses, the invented on like an MHC molecule, and if you've got
Yes.
like small enough parts of the small enough protein molecules, they'll just stick to the MHC. And
Mm-hmm.
then, so when you have like a
Yeah.
protein molecule that the T cells recognize, they express interferon gamma.
Yes.
And
A
so,
cytokine. A cytokine.
yeah, cytokine,
and,
yeah,
and that's sort of their response, like in the body when they see. So in the body they were right. That's
yeah,
the process of immunization, right? Like immunization gave this protein to the body in some weird way through electroporation, and in turn the body presented pieces of that protein to the immune system, and those pieces short because antibodies they just need a little piece, a little
yeah,
short piece to recognize. So other cells recognize those short proteins, the T cells, and the ones that and based on a variety of different factors, pool of those cells are chosen to remain around, right? Because like they're
yeah,
the good immunic cells, and so now we're crushing up the T cells are, and we're asking what is the part exactly of the protein that these things bind to.
Yeah.
And they do it through a very complicated method. Well, because they're huge. There's a lot, like they're very long, hundreds of amino acids long, right? And we need to find the 15 mer stretch that the antibody binds to. So they do it by doing overlapping pools.
Yeah.
So they basically have like, it's like if you match pool A and pool C, what's in common between them is the thing that is the epitope that it matches. And so they expose the cells to these epitope pools. The ones that wreck something produce the interferon, and actually very similarly to an ELISA, the interferon is captured by antibodies and then developed into spots. And so the actual visual like outcome of this test is that they have like a huge array of circle discs that have spots in them, and the more spots, cells are responding to the pool of antigens, and then by correlating the wells, the different pools that have the most spots, they then narrow it down to the exact pool. And so they're using three here. They're using one from the COVID-2. So again, they're really diving down. They're saying like possible that the antibodies produced by this vaccination strategy recognize a tiny piece, any of either our SARS-CoV-2 spike, classic SARS, or MERS. So this is great, right? Like this is like super detailed version of what we saw in, where was it? It was like all the way up at 3 in figure 3, right? Super detailed version of figure 3. Plus they threw MERS in to show that cross reactivity.
Yeah, MERS has got nothing. I'm so glad they made this graph just because it's an empty graph. Yeah, SARS has got some parts of it activated. That's interesting. I'd be interested to see what the overlap between those two pools are.
Yeah, like what's the epitope that SARS is being recognized for, right?
Yeah.
Did they talk about? They didn't talk about the discussion. I guess they didn't. I don't read it.
I don't remember reading it, but I mean, it could be that they skimmed it over. It could be that I skimmed it over. I don't want to put that on them just in case that they did do it. So yeah, and so they had these pools, and then they there's another graph in the next figure that was very interesting where they looked at the epitope mapping matrix for SARS-CoV-2, where looking at kind of which pools were getting the most reactions.
So this is their analysis, right? Like because now that they've done all those pools, now that they can go in and they can say, I guess they do a lot more peptide pools than what they're showing, right? Because there's only 5 pools in figure 6, but in 7, like you can see that they've done 30 pools, right, in order to do the full epitope mapping. And so the epitopes. So from this, you'd say the epitopes. Oh, so SFC is spot colonies.
Yep. Yeah, we need to have those spot forming colonies. Scientific terms.
Or spot forming. I don't know. Anyways, those are the dots, right? And every dot presumably represents a cell that's responding to one of those epitopes. So from this, like we don't know what's in these pools, but the analysis goes the epitopes that are in common between pools 6, 26, and 27, and 30, right? Some common are likely the epitopes that are being recognized on the full-length protein. And so they then show us what those commonalities are, and then where they map out to the whole sequence of that protein.
Yeah, so like quite on the receptor binding domain, which is a good place to have things reacting to, and then something that's on the S2 chain as well, and which it's interesting. I mean,
like nothing. It's like it is all around that receptor binding domain, which I mean, this makes sense. This is why there isn't cross-reactivity to the MERS spike or the SARS-CoV spike, because like if the specificity is clustered around this region, and we know that those regions are different between the different viruses, then the antibodies won't recognize that
Yeah,
area. Yeah.
yeah, and that's why
Why? Yeah.
yeah, that's why
I'm trying to.
we looked quite a lot about how different those were, and we can
Mhm.
see right now how the effect that is happening.
Yeah.
So in television terms, that was foreshadowing. So
And and papers do that a lot, right? Like
yeah,
that is the way scientific papers are written,
yeah,
right? They kind of draw you along, right? Like the story they want you to see. They
yeah,
foreshadow into what we end up learning at the end.
yeah. I mean, even scientific papers rely on storytelling. It's just a lot
Hmm.
more subtle than what you see in other places, and yeah,
Yeah.
the characters are a bit less one-dimensional, a bit more.
But also like it's not just and also yes, that's how we learn what the scientific paper is trying to tell us, but we also can disbelieve it at any point, right? Or we can
Yeah,
say, I don't think this information is useful. Right? Like
yeah,
at the end of the what they want to do is they're working towards, building a vaccine for people, right? And
yeah,
what we want to see is some efficacy. So these are good steps towards that. And
yeah,
as I said, like I'm craving to see like are these the antibodies that end up doing things? Because what if like
yeah,
a region that you want antibodies directed towards? Um, but
yeah,
I think a lot of people have done the thought about this because a lot of people have thought about this already, and we probably just haven't read the right background literature to speak more broadly about it. But there are other other coronaviruses, and there are other there are SARS vaccines in development. There's lots of old literature on that, and you can see like the antibodies that are developed against those, whether or not they were against the same region and if they protected. So like these are other things that we can look at to try to make a
yeah.
decision.
Yeah, I mean it's so. I mean the final part of that paper is just the table that illustrates the kind of. I think that basically we've covered the paper. Um, but yeah, it's. I mean, again, I'm kind of disappointed they didn't do challenge studies as you mentioned, and it's kind of interesting that they're going straight to that basically now with the with. I
Yeah,
mean,
yeah. Well, they won't challenge people in the phase 1
oh yeah,
clinical. They mention in the paper that they are going down the path of doing animal challenge.
yeah,
But I think because there's timelines here involved in terms of drug approval, and like they want to have their safe. So phase 1 clinical, that's to get your safe dose, right?
yeah,
So they're trying to find the safe dose to give, and then they'll figure out how to give an effective dose. So they can do those safety studies in parallel to figuring out like what their efficacy looks like in animals. And then hopefully their hope is, right, by the time they get to phase 2, they'll have all that information ready to make decisions on the phase 2 trial.
yeah,
And of course, if any of those things comes out bad, like you can't give enough in the human safety, right, that to sort of like correlate with what they see effective in the animal study, that will mean phase 2 is over
yeah.
before it even.
I mean,
So yeah.
yeah, that's the thing that like, for these sorts of studies, like size does matter for animals. When you're doing like a needle injection into a small animal and using the same needle for humans, it's not going to have the same kind of dose spread.
Oh yeah, yeah.
So yeah, so we just trying to like ensure that we do have. So I mean, that's why it's important to look at larger animals, not just for relative to humans, but also just looking at the size because size does matter apparently. Um,
Yeah.
yeah,
I mean, in biology, certainly, right? Like thinking about,
yeah.
yeah, like concentrations and stuff, all that changes when you size up.
Yeah, I mean, I do feel like in terms of ethics, they probably would have wanted to have like anesthesia or something like that for their animals because, again, intramuscular injection to a mouse with an electrode for 30 seconds. I'd rather, I mean, just even from an operator perspective, I'd want that mouse to be asleep when I'm doing that injection. But I
Yeah.
mean,
They don't say it.
but
They don't say.
yeah, they say. I mean, they don't say that whether they use any anesthesia or not.
Yeah, they just say it happens.
Yeah, so like there's
These
so I
shows
mean, I feel like kind of then a little bit more should be like is required.
sure.
Otherwise, like I mean, even from like a selfish human perspective, you want your operator not to be covered in mouse bites, but
Yeah.
from an empathetic perspective, you don't want to be that unnecessarily cruel to an animal.
I mean, that's
Um,
also important inside of communicating scientific results, right? Like you want to see that they've done all of
yeah,
their experimental design up to the ethical standards that we would normally see them.
Yeah, I mean, like the Lost episode where we were talking about the primate study, where they talked about all the toys that they gave the primates. That was
Right.
actually a really good example of of being transparent about your ethics.
Yeah.
So maybe it's not a case that they, because a lot of times people don't think that since it's not scientifically relevant, they decide it's not important to talk about,
Hmm.
even though they do it.
I mean, you know, like we're talking about papers, we're trying to find a different audience to speak about papers. Other people are looking at papers to try to read them. You know,
Yeah,
like having that information in them is really presenting science as more than just going after those answers, right? But also
yeah,
all the steps, all the rigorous ensure that when you execute those experiments, they're both the data you want and are ethically compliant.
yeah. Just
Yeah.
showing that, not just like showing like that we get the answers, but we get them in the right way through the right methods. I think that's really important.
Yeah,
Um,
totally, totally.
yeah,
Yeah, so as we
uh,
said, this is in phase one, and they
yeah,
they said that they're going to also do other animal studies. We don't have any functional understanding
yeah.
of this,
I mean,
but
yeah,
we have a good indication it's specific for the protein they say it's specific for.
yeah, and it's yeah, and I mean, we'd hope that like kind of, I mean, yeah, hopefully we'll have good enough antibody responses, but it's something that we need to see more of. Uh, and we need to know whether it's the right antibody responses. I mean, I'm not sure whether based off this paper, I'd have thought that in any other situation, if it wasn't so such as in a different situation, this would have gone through to like phase 2 trials.
Oh, for into phase one, I'm not sure.
Phase one,
Yeah,
oh
I feel like
sorry,
part of that is the rush.
yeah,
And
more
like
preclinical.
it's not totally crazy to think that they're trying to figure out the dose, the safe dose, or they have efficacy. No efficacy. Is that my thought? Again, this is another thing. There's even with this like super fast and the decisions people are making. So it's possible that they already have some preliminary positive published yet, but I don't know.
Possible. But anyway, that's been interesting. I hope you might read along with us. Um, yeah, and
Send us
we'll,
paper suggestions. Tune.
yes, send us paper suggestions. If you have a
Yeah.
paper, hit us up on MyWJC or like any of our Twitter handles as well,
Mm-hmm.
and then we can talk about them next week on the show. And
So
yeah,
yeah,
it's a
we'll see
pleasure
you
reading along.
next week.
Yeah, see you next week.
Yeah.